Hello, my name is Jose Yakiman, and today I will be speaking about HIV. What exactly is HIV? How does HIV affect our bodies? What exactly does it do to our bodies and immune system? Understanding the importance of CD4 cells. I'll get more into CD4 cells as I get more into what HIV does. Um, the 7 stages of the life cycle of HIV and what medicines are used to prevent it or stop HIV from spreading. So, what is HIV? First of all, HIV stands for Human Immunodeficiency Virus, and nobody really knows where it comes from. I mean, according to scientists, the type of chimpanzee in West Africa was the source of HIV infection in humans because they believe that the chimpanzee version of that virus, which is called simian immunodeficiency virus or SIV, um, was mutated when and transmitted to humans when we were in West Africa over the decades. The virus slowly spread across Africa, and we came into contact with chimpanzees who we hunted for meat and came into contact with their infected blood. So HIV is supposed to be a mutation of SIV, which is the same or similar immunodeficiency virus, only it's called the simian immunodeficiency virus instead of human immunodeficiency virus. Um, just if you want to picture HIV, this is a picture of a infected cell. Now, going inside of that cell, we can see HIV's envelope. We can see that it has many different parts just like any other cell. We have the HIV envelope on the outside, the HIV RNA, which is essentially what will be turning into HIV DNA, and that is going to be very bad for our bodies. The HIV capsid, which is HIV's core, which contains the RNA. The enzymes inside of the capsid or the core of the HIV are right here are the enzymes, and those carry out processes in the life cycle of HIV, which I'll be getting more into depth when we do that. Here's a closer up picture of it. I do not have much better materials to use at the moment. Um, RNA is its genetic material, and it's glycoproteins. Those are proteins or spikes embedded in the envelope, which is the envelope is essentially this part on the outside, and all these green little things right there, those are the protein spikes. That is the HIV glycoproteins. So, what does this do to our bodies? Well, HIV attacks and destroys CD4 cells in the immune system. CD4 stands for cluster of differentiation 4. These are a type of white blood cell that play a huge role in the immune system. Um, these blood cells essentially help protect the body from infection. Your CD4 cell count gives you an indication of your immune system's health, which is the body's natural defense against pathogens, infections, and illnesses. HIV uses these CD4 to multiply throughout the body. It essentially takes advantage of all these CD4 cells that we need for our immune system so we can be safe from diseases and illnesses and pathogens, and they take over the CD4 cells and multiply throughout your body, which will create a huge threat as our immune systems are threatened. So are our lives. There's diseases everywhere. CD4 cells are extremely important. A normal person will have about 500 to 1500 CD4 cells. Medicines used to stop HIV can potentially stop HIV, so a human can have 500 CD4 cells and still be able to live healthily. So it's very important that people living with the disease, living with the virus, or potentially catching the virus sexually transmitted from somebody. As soon as you find out, you go get the medicine, because there is medicine that will stop it, and I'll be talking about that in just a little bit. So of course, the higher number of CD4, the better. Anybody with less than 500 are at risk to developing serious illnesses. And CD4 cells, also called T helper cells, that is where the virus attaches itself first, and it releases HIV into that cell, that CD4 cell. Then HIV inside the CD4 cell begins to change the genetic material of the cell so it can enter the nucleus of the cell. Once it enters the nucleus, which is the main control center of any cell, or most cells, the main control center, it takes control of the main control center. Essentially, the infected T helper cells, or CD4 cells, produce more HIV proteins that create more HIV particles inside the cell. So finally, the cell begins to release these new HIV particles into our bloodstream to begin infecting other cells and quickly begin to spread throughout our immune system. That is simple terms for you. But HIV has 7 stages to its life cycle. As you can see here, 1, 2, 3, 4, 5, 6, 7. Now, because the picture is not so clear, I'll essentially explain what's going on. This right here is a CD4 cell, and these red parts are the virus entering the cell and leaving the cell, as you can see. So, first step of HIV is binding. HIV binds to receptors on a CD4 cell. If we look up here, that is where it's binding to the CD4 cell. The HIV envelope and CD4 cell membrane fuse together, which is how HIV enters the CD4 cell. So if you look right here, this is where it is fusing together, the CD4 and the CD4 cell membrane and the HIV envelope. Then inside of the CD4 cell, here, if you can see, that's HIV begins releasing an enzyme called reverse transcriptase. This enzyme begins to change the DNA of the CD4 cell. HIV RNA, as I showed you before, becomes HIV DNA, which allows the HIV DNA to enter the nucleus of the CD4 cell and combine with its cell DNA. And here, inside the nucleus of the CD4 cell, HIV releases another enzyme called integrase. Integrase is used to insert HIV DNA into the CD4 cell. HIV then begins to use the processes of the now infected CD4 cell to make long proteins. If you can see this blue and red here, to make these long protein chains. And these protein chains are going to begin multiplying or replicating to create more HIV. That is called replication right here, the 5th step. So we started with 5 steps. We're on step number 6 right now, assembly. Now new HIV, RNA, and proteins move to the surface of the CD4 cell to create an immature, non-infectious HIV. So at this point, these cells are not infectious. It's HIV, but it is not infectious. It's still immature. Step 7 is when this non-infectious HIV that has been formed is now pushed out of the CD4 cell, where it releases another enzyme named protease. Protease breaks up the protein chains in the immature virus to create an infectious HIV. Now, after that, the cycle will continue. Over here you can see we have 7 steps. Step number 1, binding. That's when the HIV binds to the receptors on the CD4 cell. Step 2, fusion. That's when the envelope of the HIV and the CD4 cell membrane fuse or join together. Step number 3, reverse transcription. That is inside of the CD4 cell when the HIV begins releasing these enzymes named reverse transcriptase. That's where the name comes from. Inside of the nucleus of the cell is where HIV releases another enzyme called integrase. And then it begins to replicate these chains, these protein chains, so they can start assembling more, multiplying into more. And then it starts releasing protease into our bloodstream. Or it starts releasing protease into the cell, which the CD4 cell is now excreting, or the HIV infected CD4 cell is now excreting into our bloodstream these immature, becoming mature HIV infected cells, and the cycle keeps multiplying. Now, if you look at all those steps, step number one, that's where it binds. That's where the medicine comes in. There's medicine that can stop the HIV life cycle at multiple steps. Step 1, 2, 3, 4, and 7. That's when it binds, when it fuses, when the reverse transcription happens, the integration, and the budding. So there are 7 steps to the HIV cycle. We can stop 5 of them, and so that's why it's so important to get yourself checked out if you do have the disease. There's 5 different things that can stop it. Well, there's more than 5, but there's 5 different phases of the life cycle that can be stopped. To stop binding, once again, when HIV attaches to the receptors of the CD4 cell, that is called CCR5 antagonist and post-attachment inhibitors. Those are the 2 medicines that can stop the binding. Then there are fusion inhibitors that can stop the fusion phase of the HIV life cycle. Then there are NNRTIs and there are NNRTIs and NRTIs, which can stop the reverse transcription. Integrase inhibitors can stop the integration, and protease inhibitors can stop the budding. The budding is the last phase where the protease breaks up the chains in the immature viruses, creating the mature infectious viruses of HIV. And then that'll eventually lead to AIDS, which so far there's no cure, but it is, according to research, there may be a cure soon. Thank you for listening to me speak about HIV, about what HIV does to our immune system, about how HIV takes over our CD4 cells, the very important white blood cells that we contain, and it just multiplies in our immune system and starts getting rid of our CD4 cells until we're prone to all kinds of diseases, illnesses, pathogens, and etc. Thank you.